Hello friends, I am here with my channel Solving Questions with Gauri ma'am. Here we solve questions from various MSc entrance examinations. So the question for today's video is from JNU Entrance for Biotechnology 2018. A major class of non-translated RNA function in regulation of gene expression by base pairing to mRNAs. The options are rRNA, antisense RNA, regulatory RNA, or small nuclear RNA. So let's solve this question today by looking at every option individually. Yes, starting with the first option. First option is a rRNA. This is a ribosomal RNA. This is present in the ribosomes which are involved in protein synthesis. So these do not have any role in gene regulation. When eukaryotic ribosome is there, it is having 18S, 5.8S, 28S and 5S rRNA in its ribosome. Whereas in prokaryotic ribosome, there are 16S, 5S and 23S rRNA are present. Now moving to the option B and option C. Option B is a antisense RNA. So this is the RNA which is involved in a gene regulation. The example of antisense technology is flavor saver tomato. What has been done in these tomato? Polygalacturonase is the enzyme which is thought to cause the ripening of the tomato. So, to inhibit this ripening or to prolong this ripening, the antisense RNA is used against this enzyme. And so, the expression of this enzyme is inhibited and the shelf life of that tomato is increased. So these are also involved in a gene regulation. Secondly, the option C is given as a regulatory RNA. Under this, we can find siRNA, that is small interfering RNA, miRNA, microRNA and piRNA, PV interacting RNA. So these types are also involved in a gene regulation. These are a small silencing RNA having a 20 to 30 nucleotide long. Here we can call this antisense RNA as a type of regulatory RNA itself. Yes, now moving to the last option, option D, that is the small nuclear RNA. These are small RNA which are involved in a RNA splicing. In case of eukaryotic cell, genes are first transcribed into mRNA which is having introns as well as exons. So out of these, introns are not required for the formation of proteins. So when heterogeneous mRNA is spliced and introns are removed, that function is carried out by these RNAs. When these small nuclear RNA combine with some specific protein, that formation is called as a small nuclear ribonucleoprotein, or it can also be called as a SNRPs. So these are involved in RNA splicing and secondly in the maintenance of telomeres. Hence, do not have any role in a gene regulation. So, with this discussion, what we find is rRNA is there present in a ribosome and that is involved in a protein synthesis. So, do not have any role in regulation of gene expression. Secondly, antisense RNA and regulatory RNA. These two are. So here we can call antisense RNA as a type of regulatory RNA. These are also come under the category of small silencing RNA. So these are involved in a regulation of gene expression, right? So according to me, the both B and C can be an answer for the question, but in that also more appropriate answer would be a option C. Okay, and small nuclear RNA, we have seen that are SNPs and that are involved in a intron splicing. So again, that do not have any role with the gene silencing. So the option C can be called as a correct answer for this question. So if you like my videos, please do not hesitate to like, share and subscribe for my channel, and also to hit the bell icon. Thanks for watching.